Okay, good people. Let's continue on here. We're looking at the spermatogenesis, the genesis of sperm. So, the creation of sperm in the testicles, right? All right, so let's get moving with this, right? Sperm are called spermatozoa in science, all right? Sperm is the short form of spermatozoa, all right? They're produced from a type of stem cell, it's an adult stem cell. So stem cells are undifferentiated, they're unspecialized. Okay, they have yet to get their full specialization. So adult stem cells are cells that can create different cells. Like we have stem cells in our bone marrow, which create all of the bloods of the immune cells, all right? And that one stem cell can produce any immune cell that's required. This stem cell produces sperm, all right? So they're called spermatogonia, all right? And this is by way of mitosis and meiosis, and we'll get into this. You're like, what do you mean? Isn't it meiosis? Yes, but we'll see how that mitosis piece fits in. Okay, all the red parts are needing to be added to your workbook. All right, or fixed. Okay. So within the testicles, we have what are called seminiferous tubules, and you can see within this diagram here, don't have any. Okay, so there we go. Within this diagram here, there's a whole bunch of tubules within the testicle itself. They all merge at the top, and then here we have the epididymis, and you can see how all the tubules connect to the epididymis, and you can see one tubule being stretched out here and cut, and so we can kind of look at it, and we will in this diagram here. So we'll come back to that in a second. So seminiferous tubules, all right? Now, where immature spermatocytes are released. So immature spermatocytes are released into the tubules, and they swim their way to the epididymis, where they further mature. So the maturity is that they're just not ready to swim the full distance to the fallopian tube. All right, they're going to need to grow and get stronger, basically. All right, so now, within the seminiferous tubules, all right, and this is a little cross section of a tubule, okay, you can see here the spermatogonium on the very outside of the tubule, all right, spermatogonia, okay, and then besides spermatogonia or helpers of spermatogonia are called their Sertoli cells, okay? These are a nurse cell, all right, but really what they do is they stimulate the spermatogonia to undergo meiosis, okay, and then they nourish developing sperm cells. Okay, so those two roles of the Sertoli cell, to stimulate the spermatogonia to create sperm, and then to nourish developing sperm cells. We're going to take a look at all of this right away after we look at this. All right, so what's this mitosis meiosis piece? So we have a spermatogonium here, okay? The spermatogonium, all right, will first copy itself, all right? There should be another arrow coming from here to here, all right? It will first copy itself, it will make a clone of itself, okay? Then one of the clones will go on to create sperm cells. Okay, we call this, it should say here, so you should write this in your book, the primary spermatocyte. Okay, the primary spermatocyte and the spermatogonium are the same cell. Remember, they're clones of one another. This cell, the primary spermatocyte. The primary spermatocyte will undergo meiosis, and then because the spermatogonium has copied itself, this cell here, the spermatogonia cell, will do the same thing later on. It will undergo meiosis, it will create a primary spermatocyte. Primary spermatocyte will create sperm. Okay, so what's the point of all this? It is to recycle the spermatogonia. Otherwise, we'd run out of spermatogonia very fast. Okay, so this is how the spermatogonia or the stem cell, remember it's a stem cell, is recycled. Okay, primary spermatocytes undergoes meiosis 1 to develop into secondary spermatocytes. Secondary spermatocytes undergo meiosis 2 to create what are called spermatids. Spermatids will then differentiate and grow into sperm. Okay, we don't need to talk about chromosomes and all of that. You just need to know your ploidies. So ploidies, remember we have a 2N for the primary spermatocyte. Meiosis 1 is the reduction division. We have 1 N cells, haploid cells, that also divide to create still more haploid cells. And of course, sperm are haploid cells. They are a 1N cell. Now, here's a note at the bottom, and we'll look at this in the next slide here. These Leydig cells, okay, they're called also interstitial cells. Okay, we're just going to wait for this thing to go away here. There we go. All right, they're outside the seminiferous tubules. They're still within the testicles, okay? They are the testosterone producing cells, okay? And we'll get into the function of testosterone in lesson 3 here. Okay, so, here we have it all. All right, I put the arrow here and wrote in primary spermatocyte. We already went through this, so let's keep moving. So here's a cross section of a seminiferous tubule. Okay, we can see the spermatogonia, which are on the outside, and then look on the, on the inside wall of the spermatogonia, and look on the outside of the tubule itself. Okay, this is the interstitial space, and you can see Leydig or interstitial cells here, and these are the cells that create testosterone. Okay, a lot of this you don't need to know, most of it. So only what I point out, the rest of it you can cross out. So Leydig cells, and these are also called interstitial cells. So you need to know both names, please. Don't worry about the basement membrane. Don't worry about the myoid cell or the rough ER, or the residual bodies, or the BTB, or the mitochondria, the lysosomes. Sertoli cells we'll look at here, the BTB here as well, no worries about it, all right? So let's look at this from a different perspective, all right? Spermatogonia, they are, okay, what does the spermatogonia do? It's there, it will create sperm eventually. This spermatogonium has divided to create, well, a spermatogonium that we can't see, of course, because it's already divided. Let's put it in it better, okay, has divided to create a spermatogonium and a primary spermatocyte. Okay, don't worry about the B's and A's, it doesn't matter to you, all right? In fact, I don't understand exactly what they're getting at with the B's and A's, so let's just ignore it, okay? We have spermatogonia that are basically attached to the basement membrane, okay? They will undergo mitosis to create a primary spermatocyte and a, right, so here's the primary spermatocyte. Notice how as these cells start to divide, they start to migrate from the basement membrane to the middle of the tubule, which is of course connected to the epididymis. Okay, primary spermatocyte will divide to create 2 secondary spermatocytes, which are of course N cells. These are haploid. All of these cells here are diploid. Secondary spermatocyte divide to create spermatids, and of course these are N cells here. They're differentiating. All right, they create sperm, and the sperms wiggle their way out of here, these immature sperm, and they move to the epididymis where they mature further. Don't worry about histology. We're not doing a diploma, so you won't get any questions on histology. And we'll start the next lesson with maybe sperm morphology and and hormonal regulation. That's right. That's what we'll do. Okay. Cheers. Good luck with your studies, and we'll see you in the next lesson.